Hello students, good evening. So today I will discuss seed germination test. So why seed germination is very important. So first we will know what is the seed germination. So already I think you might be knowing. So this is the nothing but it is the sprouting or germination is the sprouting of seedling from a seed, either from gymnosperm or angiosperm. So whenever you are sowing a seed into the soil, so you have to wait whether it is sprouting or not. So when it is not sprouting, means your seed lot is not good. So germination is defined as emergence and development from seed embryo of those essential structure for the kind of seed in a question. It means its ability to produce a normal plant under favorable condition. We suppose you have purchased a seed from, or you have to procure the seed from different markets, or a seed agency or certified seed agency, and if under favorable condition, if seeds are not able to germinate, it means your seed lot is not good. Your seed is either dormant period or maybe dead. So that's why germination test is very important. So now we'll see why germination test is important. So germination rate of particular seed lot is a key indicator as to how that seed will perform in the field. Because if you are sowing in a small area, that's not a matter. But if you are sowing for 5 hectare land or 10 hectare land, so their seed really play big role, because you are going to lose if seed will not germinate properly. So you are going to lose a great amount of yield. So germination rate is expressed as a percentage. So every time we are calculating germination in terms of percentage. So suppose if I am telling that 90% germination rate, it means suppose you have sown 100 seeds, it means only 90 seeds out of 100 are grown. So this information is important for calculating optimum seedling rates, as well as helping to determine whether a particular seed lot has the potential to produce a good crop. So that's why when after germination test, if it's not thriving well, or maybe 20% germination or 30% germination, means that seed lot is not good. So you have to change the seeds. So that's why before taking the seeds for the larger area, first you have to go for the germination test. Now how germination test is done? So all the accredited seed testing laboratory use germination test methods. So you have to go to the testing lab, and that is prescribed by the again CFIA. So testing method used in laboratory are controlled so that test can be repeated. Good, because you have to repeat the test again and again within the laboratory or between the laboratory. Maybe in one lab, maybe due to some problem, you are not able to connect the what you call testing well. So that's why you have to test in other lab also. So conditions like oxygen, light, moisture, temperature have been standardized for every crop type to ensure that germination will take place within a specified period of time or not. Because every plant has a particular germination period. Suppose you have sown the seed, so how long you have to wait to get the radicle and plumule? So that's why you have to standardize, like you have to standardize the oxygen, light, moisture, temperature, because and that's why using a seed chamber or seed germinator, that is very important, because you can grow out seed outside also. But if you have all the control conditions like oxygen, light, moisture, temperature, so it should have all the criteria, it should have all the favorable condition for germination of seed. So seeds of planting are taken at random, then evenly spaced on a blotter, so that they are not in contact with adjacent seeds. At the end of test period, seedlings are evaluated and categorized. So after doing the test, you have to evaluate the seed and categorize the seed in different categories. So first is maybe seeds are normal, because here your those seeds have able to produce useful mature plants under favorable field conditions. So only normal, when normal is possible, because here you are going to get the mature plants under the favorable field condition. Sometimes maybe your seed lots are abnormal after testing. So maybe due to the insufficient plant structure to maintain a healthy plant, such as missing roots or shoots, Maybe sometimes you have fresh seeds. Fresh seed means seeds have failed to germinate but have imbibed water. So sometimes it looks like your seeds are fresh, but just they imbibed water, but maybe still firm and fresh and capable of germination, but it remain dormant. Not they are not going to germinate. So maybe sometimes dormant seeds. So seeds are viable other than hard seeds, but fail to germinate when given the prescribed or recommended germination condition. So you have given condition, but their seeds are failing to germinate. Sometimes you may get hard seeds. So hard seeds are that seeds at the end of the prescribed test period, the seed coats are impermeable to water. So water is not getting inside the seed. That is called impermeable to water because the hard seed coat. And sometimes seeds may be dead seeds. So seeds that cannot produce any part of the seedling. So if not producing any part of seedling, so maybe it is dead seeds. So it may be normal, it may be abnormal, it may be fresh seeds, it may be dormant seeds, it may be hard seeds, and it may be dead seeds. So these are different categories after testing. So these are you can see here. You have two photographs. So in one seed seeds are healthy, in second maybe seeds are abnormal. So now how do I use germination test results? So again, you have to go for the different testing. So less than stellar germination test result do not necessarily mean a seed lot needs to be discarded. So significant improvement to the final germination can be achieved, especially when dormancy and pathogens are the present. So you can see. So now you will see seed testing. So seed testing is the determining the standards of a seed lot. Like you have to go for physical beauty, you have to go for moisture, you have to see the germination and other how to distinguish variety. So and thereby we are going to prescribe the farming community to get quality seeds. So this seed testing laboratory is the hub of the seed quality control because seed testing laboratory only they can you are assured that your seeds are good. So seed testing services are required from time to time to gain information regarding the planting value of the seed lots. Seed testing is possible for all those who produce, sell and use because nowadays so many electricians are there, so you have to go take the seeds and go for the seed testing lab and ask the ask the authorized person to get the seed testing done. And then only you can go for the cultivation of crop in the larger area. Now we see importance. So what is the importance? Again, importance is what is the importance of seed because if seed testing is done properly, like if you going to check all the attributes in terms of beauty, moisture, germination, vigor, seed health, then you are going to get the higher yield. So quality control of seed depends upon the different seed testing protocol, which determine the genuineness of the cultivar. So these are the certain some methods how we are going to test the seed. So germination test shall be. So this is very important. You have to remember. So germination test shall be conducted with a pure seed fraction. A minimum of 400 seeds are required in 4 replicates of the 100 seeds. So 400. So you have to take 100 seeds and you have to replicate into 4 parts. So total seed will be 400, or 8 replicates of 500. Sorry, 50, or 16 replicate of 25 seeds, each depending on the size of seed and size of containers of the substrate. So you have to take the at least because sometimes what people or what farmers are doing or what students are doing, just they are taking 10 seeds or 15 seeds, and based on germination test they are determined that these are the germination percentage. But at least you have to take the 400 seeds or and even 100 seeds in 4 replicates randomly. Then you can go for the germination test. So test is conducted under the favorable conditions of moisture, temperature, suitable substratum and light if necessary. No pre-treatment to the seed is given except for those recommended by ISTR, that is called Indian Seed that is called Treatment Association. Now materials required. So just I told you substratum. So substratum means it acts as a moisture reservoir and provides a surface or medium for which the seeds can germinate and seedling grow. The commonly used substrate are sand, germination paper and soil. So see, soil is not it is an important substrate, an important medium, but generally we are doing all the experiment on the sand or either paper. So now first we will see sand. So again, the size of sand particle is important. So sand particles should not pass through around 0.880 mm sieve and retain by 0.05 mm sieve. So this is the neither large or not small. Second, toxicity. This is very important because if sand is toxic, so maybe your seed will not germinate. So sand should not have any toxic material or any pathogens. If there is presence of any pathogen found, then seeds should be sterilized. Sand should be sterilized in an autoclave. So sometimes you are autoclaving the sand so that all the pathogens will get killed. Now, method of placement. So there are again different methods like seed in sand. First is seed in sand. You have to you have to plant the seeds into the uniform layer of moist sand. And second is top of the sand. So seeds are placed into the surface of the sand. So and second is spacing. So again you have to maintain the spacing. Then third is next is D is water. So again water is a master condition is very important. So amount of water to be added to the sand will depend upon the size of the seed. Like so for cereals, except maize, sand can be moistened to the 50% of its water holding capacity. So that's why it is very important which methods or what seeds are going to use. So this is called sand method. Now we will discuss second is paper method. So again most widely used paper substrate are filter paper or blotter or towels, that is called craft paper. So it should be capillary movement of water at vertical direction. So due to the capillary movement it will move vertically, and it should be free from again toxic substances and free from any fungi or bacteria. It should hold sufficient moisture during the period of test, and texture should be such that roots germination will be easy. So that's why these are the advantage over the sand, because sands maybe because you're going to send your autoclave it with paper. Sometimes also you are autoclaving the papers, but without autoclave also it will work. So now first we will see sand. So again the size of sand particle is important. So sand particles should not pass through around 0.880 mm sieve and retain by 0.05 mm sieve. So this is the again between paper. So you have to fold the paper and between, so your seeds are germinated between the two layers of the paper, and seeds are placed between the two layers of paper and rolled in towel. So again it will start germination. You can see the figure. Now third is, so these are the different crops. So what is the substrate, what is the temperature, and what is the first count days, because you have to count you have to check what is the percentage rate. So first count in case of ID it will be 5 days, and final count after 14 days. So again these are the different crops, all the details are given here. Okay, coconut, brinjal, tomato, etc. So now germination apparatus. So again there are different types of germination apparatus, like we have germination cabinet or germination room. Then we have room germinator. You can see this is the seed germinator, and also we have plant growth chamber. You can grow the seed in plant growth chamber also, or sometimes seed germinator is also coming. So you can germinate, just put the seed in a small, there is a small pots, and you can see because that particular germinator is maintaining all the conditions required for the seeds. And third is seed counting boards. So again there are some counting boards. So this is used for accurate counting and spacing of the seed. So this consists of two plates. The basal one is stationary and top one is movable. So based on that you can go and counting the board. And after taking the sample, the top plate is pulled in such a way that the holes are in one line, so that a fixed number of seeds fall onto the substrate. It will fall. And next is vacuum seed counter. So this is the consist of head, pipe and wall, and there are plates of 50 or 100 holes which can be fitted to the head. Again last is impression board. So it is made of plastic, wood with 50 or 100 holes per pins. And here knobs are arranged in equal length and space. By giving impression on the sand, it makes uniform depth and spacing of the seed. Thank you.